The excitement as a new scientist, as any scientist, is the new idea and the what if. What if that was true? What if around the corner we've got that instead? But if you never have the courage to think about that possibility, you're never going to find it. And you're never going to be excited.
Aging. None of us can escape. Like gravity, it pulls on each of us. Why do some of us age gracefully and others don't? How do our bodies and minds experience aging at the cellular and molecular level? Why do we even age to begin with? And maybe most importantly, can we do anything about it? My name is Gordon Lithgow, and here at the Buck Institute in California, my colleagues and I are searching for and actually finding answers to these questions and many more. On this podcast, we discuss and discover the future of aging with some of the brightest scientific stars on the planet. We're not getting any younger, yet. Arne Akbar, it's delightful to see you and talk to you again. And thank you so much for setting aside time to do this podcast. I know I'm going to learn a lot today. So you're a professor of immunology, and the decision to go into immunology, where was that obvious to you from the start?
Well, when I was actually doing my PhD, immunology wasn't a subject in its own right. It was part of pathology. I did my PhD in a department of pathology in Southampton, looking at immune cells, but in Hodgkin's disease. And I was working on humans at that time and looking at human spleen, which is a major source of white cells, especially when you have a cancer there. I was up to my elbows in cells all the time. And unfortunately, in those days, in hindsight, there were really no tools to actually investigate in-depth immunology like we have now. I worked on T cells just in the era of monoclonal antibodies, where we could finally put handles on what the T cells were, rather than say, here's a T cell and here's another one that look very similar to me. But now we could actually put handles on them and say, well, this T cell is definitely a young T cell. And we could then pick up the older T cells as well, the ones that matured into robust killers. And that gave me the big interest in trying to identify later on what happened to these T cells as they got even older still.
How do you see aging now? I mean, how would you describe aging as someone who's come into the field from maybe a slightly different discipline?
Aging was something that was always inevitable, and it's a bad scenario. You got older and things became bad. In the current era, in the last 5-10 years, it's become more optimistic. Yes, you get old, but now there are more and more things that you could do to alleviate the detrimental systems, the things that go wrong when you get older, essentially. The drugs to actually block inflammation that causes a lot of problems. But now the exciting advent of being able to clear old cells away, which can then rejuvenate organ function. I mean, that's very exciting.
Super. Okay. So let's go to innate and adaptive immunity.
Well, the important thing about the immune system is that you need all parts of it to work optimally. Each part relies on the other parts that create the whole. You should think about the immune system as an army. You have the early soldiers that go into battle. You have the more specialized soldiers later on. You have all kinds of sophisticated machinery, but it all has to be coordinated. If the immune system is not coordinated, all these dangerous soldiers could turn against you. And you have the innate side is more of the early response elements of the army, whereas the adaptive side are the ones that come in later that are more specialized with all the heavy guns, heavy machinery, and the know-how of how to actually solve the problem. But you need that early protection, so you don't succumb to the challenge early on by having those innate troops which protect you until you get the more specialized troops in.
So the innate system, like that, even exists in the tiny worms that we work on in aging. When did the adaptive system evolve, and how are they connected to each other in any way?
They evolve. I think the point where you start getting an acquired system is, I'm not an expert on the early changes that happen through phylogeny, but when you get to become a fish, you can start getting adaptive immune responses.
I see.
Which is why the zebrafish is such an exciting model for people working on the immune system, because they're easy to grow, they grow very quickly, and they've got an innate and also adaptive part of the immune systems. They can talk by mediators they secrete, they can both influence each other, they can recognize different structures on each other's cell surfaces as well, on the innate side and on the acquired side. So there's a lot
Okay.
of communication.
Hmm. I'd like to talk about skin because I know that you like to talk about skin. But before that, let's just get one other definition out of the way, and that's the T cell. Tell us about the T cell.
Within the specialized troops that comprise the army, there are a couple of very important cell types. In the old days, the T cells used to be called the conductor of the immune orchestra. Nowadays, a lot of people who work on B cells, the other side of the acquired response, they think the B cells are far more important than T cells. There's always a slight rivalry between T and B cell people.
Mm.
But the way to think about this is that, you need them both. They're both important. Think about antibodies, B cells. Think about the killer cells, the cells that can actually kill tumor cells. Those are the T cells. But you need to have crosstalk between them.
All right, let's get on to skin. Now, this is something that is, I guess, going back quite many years that you've been interested in. And I guess I heard when I first started thinking about aging that, well, the skin, we see changes in the skin with age, but it's all a response to UV damage. There's nothing really happening there in the skin, but that's not the case, is it? The skin is an important organ for aging.
To be totally honest, the reason I got into skin was because I wanted to investigate human immunity. And if you want to work on human immune responses, the most accessible place you can look at, apart from the blood, is actually in the skin. If you look in the blood, you look to see what happens well after an immune response has been initiated. Whereas we wanted to look at an in vivo immune response in humans, which is actually very difficult, but
Mm,
where we started the response of, and then be able to follow the progression of that response and how different cell types come in at different times, how they interact with each other at different times. And ultimately, what goes wrong in older people in the skin? Older people get more skin cancer, more skin infections, they have
yeah.
decreased barrier function. All these things contribute towards dysfunction. And the skin, it's very interesting, but it's the most accessible part of the body in a human that we can actually investigate.
Got it. So you mentioned zebrafish as a potential model system that's exciting. Where are you on? And you've always been, I think, human centric in your research, and that's fantastic. You're way ahead of your time in that regard. But coming back to other models, where does the mouse stand, for example, which is where most of us are focusing our attention in interventions and so on?
I was still a young postdoc in the days where the initial genetic models for mice and knockouts and knock-ins came into being. And everyone was totally overwhelmed with all the things you could do with genetic experiments. And yes, a lot of good work was done. And in certain systems, the mouse and the human are very similar. If you look at all the death genes, for example, even the nematodes, as you know, the death genes are actually very similar. And so
Mm.
working on early organisms is great for certain pathways, but not for others. For example, you can't look at the acquired immune system in those lower organisms.
Yes.
And so I think the important thing to understand is where you can extrapolate, where you can't. And so working in humans, we could get to a certain point where we could follow the kinetics of an immune response. But then if we want to follow a mechanism to see what changed, we have to go back into a mouse again. But we have to find a defined system where what we're looking at is actually relevant back again to the human. So it has to be
Hmm.
cross-informative, which meant that you had to define very clearly what you could extrapolate, what you couldn't.
Yeah.
Luckily, in the last 5 years or so, we found mouse systems that very closely resemble what we're looking at in terms of mechanisms. So we're looking at a molecule called the sestrins, and they regulate a new complex of proteins that can actually inhibit the function of T cells. They're sestrin
Mm.
knockout mice, and the sestrin knockout mice work in exactly the same way as the humans in this kind of context. And these
So
molecules are
sorry,
fascinating.
they inhibit the T cell development?
They inhibit the T cell proliferation.
Okay, so does that mean that the knockout mouse is a healthier mouse?
It seems to be, funnily enough. But if you look at trying to boost an immune response, so you could vaccinate young mice, and old mice don't respond very well. But you
Hmm.
can look at vaccine responses in old and young sestrin knockout mice. They work a lot better. And you can get up
Fascinating.
to
Wow.
up to 20-month-old sestrin knockout mice that look totally healthy compared to wild type mice. That's anecdotal. But
Wow.
you don't lose them early. They're there.
That's incredible. I mean, I mean, it does obviously pose the question, what are they there for? I mean, there must be a positive benefit to these proteins?
So you touched on a very important point, which is looking at signaling pathways in cells. Everyone investigates pathways in isolation from all the other pathways that are going on.
Mm,
The interesting thing about the sestrins is that you can turn them on by senescence. Aging in a cell will turn on the sestrins, in a T cell, that is. But
hmm
also, nutrient deprivation will also turn on the sestrins. And once you get to the point where the sestrins are turned on, the downstream pathways are very similar. This means that you can have convergence of different signals upstream that lead to the same downstream effect, which is blocking functions of a cell, but
hmm
initiated by different means. Either
hmm
aging can do this, or also lack of nutrients can do this. And you block the cell from dividing, which is very energy consumptive.
And you mentioned senescence, and we should go there actually and think about this, because it's clear that senescent cells are both beneficial and detrimental at different points in our lives, and in surprising ways actually. And it also speaks to the challenge of targeting an aging process like senescence, that you have potentially beneficial and detrimental effects. Just unpack senescence for us, if you would.
Senescence is thought to be an anti-cancer mechanism. So if a cell divides too much, eventually there will be mutations in the DNA that could make it very, very bad for you, because these cells could grow out of hand and become a tumor. And senescence is a process whereby you actually stop a cell with damaged DNA from dividing. So it's thought to be an anti-cancer mechanism. But the conundrum for the immune system is that you have challenges all through your life from the same bugs over and over again. And each time you have a challenge, you actually expand the cells that are specific for that organism. You don't maintain large numbers, otherwise your body will blow up with new cells each time you have a new infection. And
Oh,
so
okay.
at the end of a response, you actually cull most of the cells, the memory cells, the ones that remember the infection. But you have a few there that can actually recognize and respond more quickly. That's the whole concept of immune memory. That's what vaccination is all about. You remember the response and respond more quickly to boost the response. And so the conundrum is that you have to get cells to divide when you meet the infective agent. But there's this limit to how often a cell can divide because of the anti-cancer properties of senescence. And so if you're looking at senescence, yes, it's good for you to stop cells growing too much. But from the perspective of the immune system, if the cells can't grow too much, you have potentially decreased immunity. And how can you overcome that? And that's been one of the challenges that we faced.
So then there's this concept of sterile inflammation that my office is right next to Judy Campese's office, so I've heard that phrase a few times in my life. And so how does this interact here? Because you're telling me about the yin and the yang about cancer and infection, but there's also this idea that you don't necessarily need to have infection to see inflammatory processes take off and then drive other aging pathologies. Is
Yeah,
that right?
yeah. I think it's been known for a long time that as you get older, there are more inflammatory mediators floating around in your blood anyway. The question is, where do those inflammatory mediators come from? And are those mediators actually bad for you as they accumulate with aging? These mediators can come from a variety of sources, one being defective barrier function. So the gut as a barrier, keeping the bugs, most of the bugs out
Mm-hmm.
from the inside of your body. That barrier breaks down a bit. So you've got leaky gut syndrome. Another barrier is the skin. The skin works very well at keeping things out. But when you get older, that barrier breaks down. You might get other things coming in as well. If you put on too much weight, the fat cells that you accumulate, they're actually very inflammatory. So that's another source of this sterile inflammation.
Mm.
And there's also the inability of your clearance systems in the body to get rid of misfolded proteins, get rid of debris. You get kind of clogged up with all this. It's like looks like all the people who do all the garbage collection going on strike essentially when you get older, and that all creates inflammation as well. And also senescent cells, and your neighbor in your office, Judy, is one of the perpetrators of this concept. Senescent cells that accumulate when you get older also secrete a lot of inflammatory mediators. And this is called the senescence-associated secretory phenotype, or SASP. But no one really knows whether the SASP or this inflammation is bad for you. And I'll tell you very briefly about how we address this.
Hmm.
We found that in the skin of older people, if you just challenge it just with a bit of an injection, not even the antigen that causes the immune response, just a bit of
Yes.
perturbation gives you a lot of inflammation in the skin in older people, not in younger people.
Hmm.
And this was correlated to their ability to respond to an immune challenge that you gave in the skin, an immune challenge to which the older people should be already immune to, like the chickenpox virus, for example. So we gave them a little chickenpox-like stimulus, and they couldn't respond, even though they'd had chickenpox in the past.
Yeah.
And so we had a hypothesis that if maybe we could block inflammation in the older people, we could reconstitute the immune response. So saying that this baseline inflammation that increases with aging, is it good or bad? If it was
Hmm.
bad, if we blocked it, maybe you could actually reconstitute the immune response. We use an anti-inflammatory drug that was donated to us by GSK, so it wasn't any conflict of interest there. And we
Yeah.
carried out the study. And so we treated older people with this anti-inflammatory drug just for 4 days, just tablets, 2 tablets a day for 4 days. It's an
Hmm.
oral inhibitor. And we found that if we blocked inflammation in older people, and the inflammation wasn't just in the skin, it was everywhere, we
Hmm.
could actually boost the skin response in those individuals.
Hmm.
So proof of principle that in a human, you could temporarily block this high baseline inflammation to actually enhance the immune response. And in our studies, a lot of that inflammation, at least in the skin, was coming from the senescent cells.
That's fantastic. That's really amazing. Does that also say that the senolytic drugs that are being developed, we've got something like over 100 companies now targeting senescence. Are you optimistic then that these things could be really beneficial?
I'm optimistic. You have to be, as a scientist, you have to be optimistic. Otherwise, we wouldn't be getting out of bed and going to work. I'm optimistic that there's an avenue ahead that could be beneficial, but there's still lots of things to know. Like, how long do you have to block the senescent cells for before they come back again? Probably the
Yes.
same with any drug you use. You could say the same thing from an anti-inflammatory drug perspective. How long do you need to do it before the problems start again? What we haven't explored yet are what the long-term side effects might be. Because at the beginning, you said yourself that senescent cells could be bad for you, but also they can be good for you in certain contexts.
Hmm.
What happens if you block it for too long?
Yeah.
And so you could get rid of senescent cells as one strategy. You could also block the detrimental substances that those cells secrete, the inflammatory mediators, which we've done. And this treatment didn't kill the senescent cells, you just blocked what they secreted for a while.
Hmm.
And what is good about blocking temporarily the inflammatory mediators is that you open up a window to allow the immune system to work. So the way you should think
Hmm.
about this is maybe you can let the immune system work in an older person during cancer therapy. Maybe you can get the immune system to work better by blocking inflammation before you vaccinate someone. But you don't want to block forever in case it's bad. You can just open up a window of opportunity to let it work.
Huh. We're seeing like parallel situations even in nematodes where we're ramping up the autophagy, for example. This is work from Julie Anderson's lab. And again, it's one of those things where you don't want to chronically be doing this because there's downsides. It's going to be toxic eventually because it starts to interfere with normal processes. But if you ramp up autophagy for a short period of time, you see all these phenomenal benefits in neurological disease models and proteostasis and everything else. So it seems to be an emerging theme in aging that you want to go in, fix the damage, get rid of certain cells, and then get out again.
It's like having a bad knee and having a steroid injection. It'll last for a little while, but then you need to get another one at some point.
Could we get back to let's go back to the cell biology a bit and some of the work you've done. So you've looked at uh, intracellular molecular pathways that are involved in processes like senescence. So maybe you could talk about that.
There's one pathway we've been very interested in, which is looking to see how nutrition and senescence signaling, so senescence comes from DNA damage essentially, how these pathways can converge to give you the outcome of decreased proliferation. And the reason why that's important is that you need proliferation in the immune system. Each time you see the same bug over and over again, you have to increase the number of your cells that are responsive to that organism. One thing we were very interested in is an enzyme called telomerase. So if a cell can upregulate this enzyme, you can extend its ability to proliferate. Otherwise, you just stop proliferating because you shorten your telomeres. And that's what telomerase does. It extends your telomeres. And we know that in T cells that are towards their end stage of their life, they shut down their ability to make telomerase. My group was involved with a lot of this work for many, many years. And one question we asked is whether a cell could inherently turn on telomerase again, or is it shut down forever? What we learned about the sestrins and this pathway was that if you were to block this sestrin signaling pathway, you could actually turn telomerase on again. And you could turn on proliferation again in the cells. And you can turn on other functions of the cell as well, like cytokine production, etc. This implied that in at least T cells, the senescence or the aging process is something that's reversible.
And what's the connection to the P38 MAP kinase function that you've also studied extensively?
The sestrins are unique in that they organize a complex of 3 MAP kinases. So there are 3 MAP kinases people normally work on. P38 MAP kinase is one, which you mentioned. There's MAP kinase, and there's also junk MAP kinase. They do different things, they're all very important, but we showed for the first time in one single cell type in a human, which is the T cell, the sestrins coordinate the activation of all 3 MAP kinases. And this new molecular complex that we discovered, he showed that the sestrins are bound together in a complex, all 3 MAP kinases. And within that complex, if you want to boost proliferation, ERK, junk, and P38 can be involved with that proliferative reconstitution. But only P38 is involved with the telomerase. So it's a sestrin complex, but through P38 that turns off the telomerase. So if you block telomerase, so if you block either the sestrins or block P38, you can turn telomerase back on again in those cells.
Historically, I guess there's always been this idea that turning on telomerase was not necessarily a good thing because it was also associated with cancers. Is that not the case in T cells?
Yeah. I think it's a situation that we mentioned earlier, that you don't want to block anything for too long, especially if it's a defense pathway. You're defending against cancer. But if you want the cells to respond and increase in number, maybe during vaccination, this could be something very useful. Temporarily let the cell go. But I think if you then take away the inhibition, the cell will go back to its default state, which is no proliferation, which is key. You just need to open up that window to let it respond for a little while. But there's one thing that we haven't actually talked about, which is very important from the immune perspective, which is the immune system is thought to be there to react against infection. We talked about that. It's there to react against cancer cells and to clear them, kill them off, etc. But the immune system can also recognize senescent cells in the body, which means that if you've got old cells in every tissue, your immune system has the capacity to recognize them and kill them. So you have your own anti-aging mechanism inbuilt in your immune system. The question is, why do senescent cells increase when you get older? What happens to the immune part that stops it doing what it's supposed to do, which is clear away all the old cells?
Right.
Which means that, and this is a paper we published recently, and Judy Campisi was a co-author of this paper, is that senescent cells have evasion strategies. And one of the evasion strategies is they can increase their expression of an inhibitory receptor that stops killer T cells from actually working. Interestingly, that same pathway is used by cancer cells. And so there's a drug called monalizumab that's out there, and it's been used for anti-cancer therapy. And what that drug does is it blocks the interaction. I'll go to detail, just tell you the names, but it's just the inhibitory pathway. You have the receptor, which is the inhibitory receptor on the T cell, and it's called NKG2A. You've got the ligand called HLA-E, which is on the target cell. It could be a senescent cell, it could be a tumor cell. The drug's designed to actually block that interaction to allow the immune system to then recognize the target cell to kill it. It's an anti-cancer drug, but the same pathway operates in senescent cells. So when you use this drug as an anti-cancer drug, you can actually get rid of senescent cells without the need to kill them directly by senolytics, without the need to just block their inflammatory mediators. You could license their killing by blocking the inhibition.
Fantastic. That's really interesting. It makes me wonder, if you had a $100 million grant issued tomorrow, Where would you focus your research on with that? And I presume that you're obviously someone who collaborates a lot with other people and gets ideas and works with other people. What sort of team would you build, or what sort of project would you go after? Is this one of the areas, for example, that you would be interested in?
Just as a subtext to that, I'm involved I'm involved as the coordinator of a UK-wide grant to look at aging of the immune system. And we're bringing in people from all walks of science, not just immunologists. You've got people working on circadian rhythms, you've got people working on nutrition, you've got people working on the physical aspects, and everything's linked to, we're trying to link everything to frailty. Frailty being the
Yeah.
inability of an older person to walk very well or to just live a nice, good life. We're trying to understand how the immune system takes part in the frailty response. Because ultimately, what you want is not just to boost your immune response. We want to actually live better and live in a healthier, more productive way as you get older. Get rid of
Hmm.
frailty. That's what we need. We know that the immune system's involved with that because there are various studies out there saying that T cells, which are old, so senescent T cells, will actually affect many, many different tissues in a negative way. But if you get rid
Aha.
of those T
Okay.
cells, then everything gets better in an older animal.
This is really fascinating because we know that something like frailty, which is measured in quite simple, straightforward ways in humans, and people are doing it in mice now and so on, we tend to think of frailty as an integrated function of like neuronal function, skeletal muscle function, all these things. And what you're saying is that T cells could be at the nexus of some of that dysfunction.
Yeah. So T cells may be at the nexus of some of those functions, but here's the challenge that I think we all face. With a lot of money, here's what we should look at. We should look
Right.
at the common mechanisms that lead to dysfunction in many different systems in the body. So what links muscle frailty with Alzheimer's, with other things that go wrong, lack of responsiveness to vaccination, for example. Don't go for individual things. We can't have polypharmacy. We all know that. But
Yeah.
what are the
Yeah.
common things that link all these dysfunctional elements together? And that's what we should target. And so that the money should be put towards trying to integrate the information to find out where they all join up and target that one thing that can affect many different things ultimately. And a prototype for that is a sestrins, nutrition and senescence linked up by one pathway.
We talked earlier on about sterile inflammation and the sources of that, and you mentioned, for example, fat cells and the inflammatory processes that come from fat. And I occasionally wonder if we just have an incomplete knowledge of our microbiome. And the kind of issues I'm thinking about here is the fact that there's now this incredible interest in the idea that Parkinson's disease, for example, originates in the gut. And we've been looking at neurological disease essentially in a silo, if you like, and now we're seeing a much more wide, fuller body view of what diseases like this are.
Mhm.
Do you see that as well? Do you think that we have an incomplete knowledge of the viruses that are probably there and are inducing inflammation that we don't know about right now?
Yeah, yeah. It won't just be viruses. It'll be microorganisms, it'll be bacteria, it'll be other things as well. Everything outside is trying to get in and eat us up, essentially. And they're all, and we're fending these things off through our life. But where does the time come where we can't fend them off as well as we could before? And
Hmm.
it could lead to many, many different things. You mentioned the link between the gut and organisms there and the brain. I've just spent a short sabbatical in Cambridge working with Menna Clapworthy's lab. And she actually was one of the first people to show that link between the brain and the gut. And something else we haven't touched on, which is actually crucial in all this. No point getting a better body that fights infection better if your brain's not working. How
Yeah.
do all the things that we've talked about, how does the immune system link in with the brain? And there's now a lot of interest in the neuroimmune axis. How do brain cells interact with the immune system? We know that there are lots of innate like cells that look after the neurons. They're always associated with neurons. They're also in the brain as well. But what is that interaction? Who's the shepherd and who's the orchestrator of what's actually happening? What are these cells doing and how do they go wrong? And how can we change that axis of interaction between those cells to make things better?
Super. I mean, I mean, if our audience was to tune back in 5 years' time and we were still talking like this, what do you think you'd be saying then about the advances that we're about to make?
Mhm. A lot of our advances at the moment are linked to two things, mainly technology. The better the technology you've got, the more you can actually do. which makes me wonder how, when I started off my PhD, going way back to our earlier parts of conversation, how we did anything at all. We had so little technology to do anything. And
Right.
the other thing is you need to have conceptual leaps. And in the senescence field, in the aging field, there've been a lot of conceptual leaps quite recently. Very exciting field for that reason. The whole idea of senolytic therapy, the whole idea of the immune system being able to attack senescent cells, that's all new. And that's conceptual leaps together with technology. We can do single cell RNA-seq now. We can look at individual cells and see what they're doing within a tissue, and that's fantastic.
Mm-hmm.
We need to better organize the relationships between mouse and human systems, but also other in vitro systems. Just coordinate. That's what we need to do. And if you gave me the amount of money that you promised earlier, and I'll be looking for that in my bank account soon,
Of
I
course.
would try to coordinate and get people to coordinate interactions between people to find out how an area of research can actually go and set its tendrils into many different areas as well, and with the interactions that go on. Microbiota, important in the brain, immune system, important in the gut and the brain. The other things as well, nutrition. What happens to someone who's starving somewhere and getting old? They're going to have double whammies, triple whammies in all kinds of different ways.
So what do you say to your young scientists at UCL? I mean, this is not the world we grew up in. We were stuck in our own disciplines, and as you say, with relatively little technology to work with. What do you say? What are the skill sets that people need to acquire now?
I think, and I say this to my groups, I'm not just saying this to you, I try to instill courage that you need to be able to think outside the box. And because you think that no one will believe you, it's not a good reason to think about new ideas. The excitement as a new scientist, as any scientist, is the new idea and the what if. What if that was true? What if around the corner we've got that instead? But if you never have the courage to think about that possibility, you're never going to find it. And you're never going to be excited.
I think that's an inspiring way to end this conversation. Arne, thank you so much. Again, I learned a lot. I didn't pick up enough immunology when I was at university, and so I'm always scrambling. But that was absolutely fascinating. Thank you.
It's been my pleasure to talk to you, Gordon.
Thank you so much for listening. If you've enjoyed this podcast, please subscribe, share, and give us a 5-star review on Apple, Spotify, or wherever you get your podcasts. We're not getting any younger, yet, is made possible by a generous grant from the Navigage Foundation. The Navigage Foundation is enhancing the lives of older people through the support of housing, health education, and human services. Our podcast is produced by Vital Mind Media. Wellington Bowler is here with me, using sign language to keep me on course and recording the podcast. Stella, who I love spending time with talking about science, as you know, is our editor, with the creative direction of Sharif Izzat and the Buck Institute's very own Robin Snyder as the executive producer. If you're listening to this podcast, you know that there's never been a more exciting time in research on aging. Discoveries from our labs are moving into the clinic to help us all live better, longer. The Buck Institute depends on the support of people like you to carry on our breakthrough research. Please visit us at buckinstitute.org to donate and to learn more.